Hello students and welcome to another lecture of genetics. In today's lecture, we will be covering two main topics. The first one is regarding to population, and we are interested in this class at looking how the alleles frequency change over time or from one generation to the other. And we are also interested in the frequency of the genotypes, okay, and how that changes over time. On top of that, we are going to try to correlate those changes in the frequency of the alleles or in the frequency of the genotypes with this concept that you probably have heard, and we are going to keep digging into this field, that is evolution. Okay, so I will say that this is one of my favorite classes of all these topics that we are studying, because I do think it is a good correlation between what we understand about genetics, what we understand about evolution, how those two things are connected, and I hope that you guys will also find this interesting. Okay? To begin with, I wanted to say that throughout this class, we are going to talk a lot about Darwin, and I do have more information about him in the upcoming slides, but the main idea here that we are interested, first of all, is to study the population. The population is defined as a group of subjects of the same species. And those subjects, they can reproduce and generate a viable offspring, a offspring that can also reproduce. Okay, that is a very generalistic concept for population, and you will see that sometimes that concept doesn't actually encompass all the singularities that we have in biology. But let's assume that here population is the group of individuals that can reproduce and generate a viable offspring, an offspring that can also generate another offspring. Darwin was not the first person, but was one of the people that observed the populations and the species within those populations. They are not fixed, meaning that they change throughout generations, meaning that the generation number one of one species is not the same as the generation number two of that species. So these changes that happen within those populations lead oftentimes to the evolution of that population. And let's think of here as evolution as this evolving process, not necessarily as a good thing, right? We tend to think of evolution just as Digimon or Pokemon, right? Where one Digimon or Pokemon evolves to something better, to something bigger and more powerful. But here, this is not the case. This is a very normal misconception that some people might have, because again, that's influenced by the cartoons that we sometimes watch. Evolution here, I want you to understand as a change that happens as a consequence of the changes in the genetic material of that population. Okay? evolution here not necessarily will lead to something beneficial, to something more powerful. Okay? Evolution, these changes in the genetic material of that species, can lead to deleterious phenotypes, to phenotypes that might not actually be suitable for that environment. All right? But let's understand here that evolution happens as a consequence of changes in this genetic material. And the genetic material, and by that I mean the DNA or the RNA, happens through mutation and changes in the allele frequencies in those populations. By alleles here, let's simplify things considering just 2 alleles in that population. Okay? And by 2 alleles, we can consider, for instance, let me get the pen. We can consider, for instance, the dominant allele versus a recessive allele. Okay? Or other types of allele as well. When I talked about the codominance, not the codominance, the incomplete dominance, right? Between the flowers, the white flower and the red flower. We saw that in class number 3, I believe, or 4. Let's just simplify with 2 alleles because that's what we get from our parents, all right? We get 1 from the biological female and 1 from the biological male. The union between our understanding of population genetics and the theory of the natural selection that we are going to discuss here in this class creates a new area in the science that we call neo-Darwinism, which is a new way of understanding the theory of natural selection. Okay? Natural selection and evolution are 2 different things. Okay? Natural selection is a way of how evolution happens, is a way to explain how evolution happens. Evolution is a theory on its own. Okay? And because it is a theory, people might think that theories are just guesses, or you think that something is. But evolution as a theory in the scientific domain, in the scientific community, is understood as something that can be scientifically proved in the lab or observed in the nature. Okay? This is not just a guess, but is actually something that we can prove that happens in the nature. But natural selection is a way of explaining evolution. They are 2 different things, but they are correlated to each other. In this class, I also want to make sure that you understand when we talk about species, right? Species are those individuals that can reproduce and generate a viable offspring. When we have the event known as speciation, we have the formation of a new species. And that new species can be formed, can be generated by events that we are going to study in this class. Those can be mutation, migration, genetic drift, or even natural selection. 2 other things are important for you to understand. One is microevolution, and the other one is macroevolution. As you can guess, micro happens at a smaller level. These are small changes that happen within the population of that species. Not necessarily those microevolutions will generate a new species. Okay? Macroevolution, as you can also guess, is something that happens at a macro level. So when these events happen, and we are going to study those events, we actually create a new species. When do I have two different species? When those individuals are not able to reproduce anymore. Okay? So for instance, you have species number 1. Throughout time, there are microevolutions and micro changes that keep happening with that species. At some point, this change that happens with this species is so great. There is a mutation, or there is a migration, or there is a founder effect that we are going to discuss in the upcoming slides, that this population now becomes two different species. And now species number 2 and species number 3 can't reproduce anymore. There is a isolation, reproductive isolation between those two species. And I know that I gave you a lot of information at these two beginning slides, at these two starting slides, but we are going to explore those concepts as we evolve throughout this class. So in this class, our main goal is to understand how, for instance, new species are created and how that is related to the genetic variation that is present in those populations. Okay, so in this first slide, I just want to reinforce this concept that we can observe variation within a population. And that variation in that population is due to the genetic composition, to the genetic variability that is present within that population. Okay? By genetic variation here, we understand the composition of the DNA, right? The DNA have genes, and those genes are simply sequence of letters, right? A, T, C, G, and all those letters that we know that make up the DNA. So what I am saying is that when we look at the population and we observe certain degrees of variation, this is what's actually dictating that variation, the sequence of letters that make up that gene, because that sequence, like I said, can come in different flavors, and that's why we have different alleles for the same gene. So the first slide, this slide here is just summarizing our biological definition of population. And remember, this is a very broad definition. And we know that anything or everything in biology has some exception, right? So you might look at certain populations and might not be able to actually fit them under this category. But generally, for the purpose of this class, we understand as a population a group of individuals, subjects, belonging to the same species. Those individuals, they share or they live in the same geographic area. And it doesn't matter how spread apart that geographic area is. If those individuals are still able to meet and reproduce, then that's the geographic area that they occupy. You can think an example about this would be the Homo sapiens, right? We are all the same species and we compose a giant population. And even though a person here in the US is physically distant from a person, for instance, in Europe or any other part of the world, if they meet, they are still able to produce, to generate a viable offspring, right? If they meet, there is a chance for mating between those two individuals. So here the third definition is actually that within that population, they can interbreed, so they can mate and produce a new generation. All right? And because we are discussing about population and species, I thought it would be important we open a big parenthesis here in this slide, because I'm gonna give you an example about dogs, and you might have heard, that dogs have races or dogs have different breeds. And I want to discuss specifically about why sometimes this concept of race is wrong and doesn't actually exist. Okay? What we are defining, or what we people usually define by race, is this genetic variation that happens within the population. So when we think about the case of the Homo sapiens, all of the individuals, all of the human beings belong to the same category, belong to the same species. So we are only one giant population occupying the planet Earth, right? What we observe, and for long was understood as races, was this simple genetic variation that happens in those populations. Naturally, race does not exist, because race was a term that was created by a supremacist, by supremacists that actually viewed other people belonging to different categories in that genetic variation as inferior, as lower, just because they were different. This actually has some relationship with the natural selection concept created by Darwin, because people would appropriate of that concept to justify that some species, or that some individuals, were better or best fitted to that environment. So because of this misconception of natural selection and what Darwin proposed, people appropriated of those ideas to justify that within the population there were people that were best suitable, that were more powerful than others, just because they looked different. And because of this misconception, I would say, the term race was created to define categories that doesn't actually exist in nature, or that can't be proved to exist, because what we call nature scientifically and biologically is just a naturally continuous of this variation that we observe in the DNA. And that is because, and that connects to my next slide, that is because within that population there is what we call a gene pool. The gene pool is all the genetic information that exists within that population. So all genes, all forms of those genes, all those alleles that exist in that population make up for the genetic or the gene pool of that population. It is important for us to understand that most populations, they will have a high degree of heterozygous happening. And that is because if we think about the two alleles that we've been working throughout these classes. So here we have a homozygous dominant, and here we have a homozygous recessive. Most of the populations that we observe in nature, this is the genotype that actually exists for most of the genes. Of course, genes that do not fit in the category of multiple alleles, okay? Why is that the case? Why would we favor heterozygous in a population? We're gonna look into this issue with more details, but just so you know, for instance, there are tons of mutations that we've learned that are autosomal recessive, right? And most of those mutations, they ended up being lethal. And because of that, people that generally carry those autosomal recessive mutations, they don't reproduce. So in a population, the levels or the frequencies or the frequency of this type of alleles are usually selected, and they that phenotype ended up not being expressed as commonly as a heterozygous, for instance. Okay, so just think about that most of the times some mutations can be deleterious when they happen as a homozygous recessive, right? We talked about some examples in other classes, and because of that, they can't actually leave a offspring. Also, it is important that we understand that this genetic variation in the population can be observed, okay? And that is due to the gene pool of those species, all right? Just like we think about the skin color in the Homo sapiens, right? That skin color, though those variations in the skin color exist because of the genes controlling the gene color. So there is a variation in the alleles controlling the skin color. The example I'm going to give you here is about dogs. Dogs, they all belong to the same species. However, due to the degree of variation in the species, and specifically because of the selective breeding that happened throughout years with those animals, we have things like this. This giant dog belongs to the same species as this tiny Chihuahua. So this is what I want you to understand from this this chunk of slides, that populations, they have a certain degree of variation, and that is because of the gene pool that exists in that population. And this concept of race does not exist in nature, because what we understand about what is understood about race is this normal genetic variation that occurs in that population. The term race, again, doesn't exist, and it was created based on this misconception about natural selection. And we are going to study what natural selection is But for now, people appropriated of that idea to justify that some people, some races would be better than others. As I mentioned, our goal is to understand how that variation in the population is passed from one generation to the other. And here we actually have a law that will describe the allele frequencies and the genotype frequencies in that population. And the model that we are going to use here is called the Hardy-Weinberg law. And I am sorry about the pronunciation of this name, but you will have to bear with me, okay? Because I do have a hard time here, so just assume I am saying the right thing. But anyways, so this model describes what happens to allele and genotype frequencies in an ideal population. Remember, the allele is the flavor, right, of that gene. Here, we are working with two alleles. The genotype will be the junction of two alleles, one that comes from the mom, the biological female, and one that comes from the biological male. So the genotype can be homozygous dominant, heterozygous, or homozygous recessive. By ideal population, we understand as a population that is large, where mating happens randomly, and that population is not subject to evolutionary forces, meaning that there is no mutation, there is no migration, there is no natural selection or genetic drift. Okay, genetic drift is also another type of evolutionary force that I did not mention here. But in this ideal population, those processes, they do not happen. Because of that, I want to go over some of the assumptions for this Hardy-Weinberg model. So here, we are going to assume that the allele frequencies in our population, they do not change from one generation to the other. The allele frequencies means how much percent I have for the dominant and for the recessive allele. Okay? Here, we are not assuming that there is codominance. We are not assuming multiple alleles. We are not assuming incomplete dominance, okay? We are just working with a normal segregation, a normal monohybrid crossing here, okay? So, first assumption is that the allele frequencies do not change from one generation to the other. Here we are also assuming that after one generation where random mating happened, the genotype frequencies can be predicted using the allele frequencies. And how do we do that? By using this formula. Here, P is the frequency of the allele A, and Q is the frequency of the allele A. One is dominant and the other one is recessive. Okay? Here, this formula equals to 1 because this 1 represents, guess what? 100%. which should be the sum of this guy plus the frequency of this guy and the frequency of this guy. Knowing how many individuals I have of this, and with this genetic background, I expect that this gives me the total population. So that is why this equation equals to 1. Here are some other assumptions of this model that we are studying. First of all, there is equal rate of survival and reproduction. Therefore, there is no natural selection. We're gonna discuss what natural selection means, but here we're gonna assume that in this model, no survival or reproduction is benefited. Okay, there is equal rate of those things happening. We are also going to assume that there is no mutation, meaning that there is no new allele created. Okay, let's think that the allele dominant is given by this sequence of letters. So G, A, T, and the recessive might be A, T, C, A, C. So there are two changes here. Mutation means that a new allele, B, for instance, is created, and the base pairs here would completely change, like this, for instance. So all this is completely different, right? This is mutation. So we are assuming that there is no such a thing in this population. Also, there is no migration, meaning that no individuals come into that population and no individuals go out of that population. Another assumption is that the population is large and that random mating occurs. Knowing these assumptions, we can predict the genotype frequency based on the allele frequencies. How do we do that? Let's look at our Punnett square. In this Punnett square, I am representing the crossing between an organism that is heterozygous with another organism that is heterozygous. Here, the A dominant equals to P. That's the frequency of the dominant allele. The frequency of the recessive allele is given by Q. Here, what I am showing is the frequency of the dominant allele and the frequency of the recessive allele. Here I'm also representing the frequency of the dominant allele, the frequency of the recessive allele. Just like we did when we did our monohybrid crossing. Okay? Here in the first square, we have our homozygous dominant. So here, this frequency can be represented by P squared, because it would be the frequency of one dominant allele times the frequency of the other dominant allele. Then we have the heterozygous, right? In our Punnett square, which here can be represented. The frequency of this heterozygous can be represented by the multiplication of P and Q. Here, because we have two of this, we can just simplify that the frequency of the heterozygous is 2pq. Okay? And lastly, we know that here we would have the homozygous recessive, which the frequency of this genotype is given by q squared. So here we are using the frequency of the alleles, dominant and recessive A's, to predict or to calculate the frequency of the genotypes. Okay? So the frequency of the genotypes can be predicted or calculated by knowing the frequency of the allele. And we are going to do this in this example. So here, I am telling you that the frequency of the allele, A dominant is 70%, which is represented here. And the frequency of the allele A recessive is 30% in that population. Knowing these frequencies and knowing the principles of the segregation, which we studied when we talked about the monohybrid crossing, we can now calculate the frequency of the genotypes in that population. So, the frequency of the genotype dominant, homozygous dominant, is 49%. The frequency of our heterozygous would be 21%. Of course, here we are summing them both, so that equals to 42%. Okay? And the frequency of our homozygous recessive is 9%. So, like I said, knowing the allele frequencies, we can calculate the genotype frequencies based on the principles of the segregation postulated by Mendel. What is important that we understand from this model is that the dominant traits, they do not necessarily increase from one generation to the next. Doesn't mean just because it is a dominant trait that this is going to be favored in the next generation. Okay? Also, one consequence of this random mating that we are assuming in this population is that the genetic variability can be maintained, right? If the individuals are randomly mating, then you maintain the gene pool of that population. And lastly, which is what I've been repeating, is that by knowing the frequency of one genotype, we can calculate the frequency of other genotypes at that locus. All right? The principles of the Hardy-Weinberg law can also be applied to the human populations. And here I'm going to use one example to explain how we can apply this law. I just wanted to mention that the captions here are doing a great job to keeping up with my pronunciation of Hardy-Weinberg, because I generally doubted that this would be translated the way I am pronouncing. But anyways, let's look at one example of how this law can be applied to humans. Okay? Here we are going to do the analysis of the gene that influences the susceptibility of the infection of to HIV-1. So HIV is a type of virus that can infect the human body, and then causes, if not treated, can cause your immune system to become weaker and weaker, right? It can weaken the immune system. So here, there is a gene, or there is a mutation associated to a gene that confers some degree of protection against the HIV infection. The gene that we are going to look is the CCR5. And this gene, CCR5, encodes, or give rise to the protein C chemokine receptor 5. So very briefly, these chemokines molecules, they are present in the cell surface of the cells of the fence of the human body. So they are associated with recognizing or allowing this HIV virus entering the cell and kind of conquering the cell. If the person does not have that molecule, does not have the chemokines, then they are more resistant to the infection by the HIV. Okay, so just very briefly, this gene codes for a molecule that stays on the surface of the cells of your immune system. Here I am showing you the gene and how the gene is organized in the chromosome. Okay, so the gene for the CCR5 actually has 4 exons. Exons 1, 2, and 3, and exons 4. So there are 4 exons in this gene. These regions right there, where there is no numbers, those are the introns. As you might record from your biology classes, from your introduction biology classes, the introns are regions in that genes that do not code for any protein, okay, or for anything, or for any mRNA. All right, so here we have those 4 exons that will code for a mRNA. The mutation happens in the exon 4. So the exon 4 is where the mutation happens. And the mutation here is a deletion of 32 base pairs in that exon. So putting in other words, the exon would be represented by many letters, right? And I'm not going to put all of them here. So you would have many bases representing this exon. What happens is that 32 of these bases are deleted in this mutation, and that confers resistance to the HIV. Okay? Very basic understanding of this mutation. So here, the mutant allele has a deletion of 32 base pairs in this exon 4 of the gene, making a protein that is shorter, which blocks the entry of the HIV-1 in the cells. Here, the homozygous individuals are resistant to the HIV infection. By homozygous, let's use the nomenclature, lowercase a's. The heterozygous, they are susceptible to the infection, but it's not as severe, or it progresses less quickly in those individuals. And here, heterozygous, we're going to represent as a capital A and lowercase a. When we look at the genotype, here's how we can represent the genotype of this population. Okay? So, 1/1 or homozygous dominant individuals, they are susceptible to the HIV. Heterozygous individuals, that here we are representing by 1/32, are susceptible, but the disease or their condition, they progress less quickly. The mutant allele, which would be represented by 32/32 or lowercase a, they are resistant to most of the strains of HIV. When we do a PCR, which is a polymerase chain reaction to detect the size of those alleles, here is what we identify. The homozygous dominant individual produces the two fragments of DNA. One fragment that has a size of 403 base pairs, and another one that produces a fragment of DNA that has the size of 332 base pairs. A homozygous recessive also produces two fragments of DNA. One that has the size of 371 base pairs, and the other fragment has the size of the 332 base pairs. The heterozygous, as you can imagine, is a combination of both. Okay? So here is how we identify a heterozygous individual. It will produce the three sizes of the DNA fragment. Okay? So this technique allows us to identify the genotype of the individuals in that population. Okay? By knowing the size of the fragment of the DNA created in this reaction. Now we get to the part that we are actually interested, because the question is, how common or what is the frequency of the mutation in the human population? I am already going to tell you that the frequency of the mutant gene is of 11% in this population. What we should not forget is the principle dictating the behavior of those alleles and those genotype frequencies in that population. So now I want to show you two ways that we can compute, that we can calculate the frequencies of the alleles in that population. Here, I am going to use a population with 100 individuals. Okay, 100 subjects. And let's imagine that within these 100 subjects, I did PCR in all of them, and I found out that there are 79 people that are homozygous dominant. I found that there are 20 people that are heterozygous, and I found out that there is only one person who is homozygous recessive, meaning that that person carries the mutation. knowing that there are two ways of calculating the allele frequencies in the or based on these genotypes. First of all, let's discuss about the counting the alleles. Okay, the alternative method is starting from the genotype frequencies, but that's the other part. So how do we do that? By method A, counting the alleles. So first of all, I need to know the number of individuals with each genotype. Okay, some from a total of 100% in that population, 100 subjects in that population, I found out that 79 have the genotype dominant, homozygous dominant, 20 have the heterozygous, and only one has the mutant allele. In that population now, I want to know what is the number of the allele dominant. Okay, here I'm representing that with 1, but I'm putting the capital A just so you can relate more easily in your head. Here we know that the genotype, let me get the pen again, that the genotype homozygous dominant has 2 alleles A, one that came from the biological mother and one that came from the biological father. Okay, if I know that I have 2 of the same alleles in that population and I have 79 individuals, therefore the number of dominant alleles in this category is 79 times 2, which equals to, oh my goodness, which equals to 158. Okay, which is represented right here. Now, how about the heterozygous? The heterozygous, we know that one allele came from the biological female and the other allele came from the biological male. Okay? Here, I only have how many individuals? 20 individuals in that category. However, of those 2 alleles present in that organism, only one is actually carrying the dominant version of that allele. So here I do 20 times 1, which equals to 20, and that is represented right there. How about the individual that is lowercase a and lowercase a? Remember, one came from the biological female, the other one came from the biological male in those individuals. Here you can see there is no dominant allele. Therefore, the number of dominant allele in this category is 0. Now the other question is, what is the number of recessive alleles? Well, you can already imagine that in the homozygous dominant, the number of recessive allele is 0. In the heterozygous, the number of recessive allele is 20, because now we are counting these other alleles. And here, for the recessive allele in the homozygous recessive, we have 2 alleles because we only have one individual. Okay? Now what we can do is sum the number of the alleles dominant and the number of the alleles recessive. Here, for the dominant allele, we have 178 alleles, and for the recessive allele, we have 22. So in total, I know that I have this much of alleles. I have 200 alleles in total in that population, which makes sense, right? If we have 100 individuals being analyzed, that means they have 200 alleles, because each one of these individuals have 2 alleles for that gene. All right, one that came from the biological female and the other one that came from the biological male. Okay, now what we can do? Simple math. Let me erase everything here. Now what we can do is, knowing that I have 178 alleles dominant in a total of 200 alleles, I divide one by the other and get that in that population I have 89% of dominant alleles. So the frequency of the dominant allele is 89%. The same thing can be done with the recessive allele. If I know that in the population I have 22 recessive alleles out of a total of 200 alleles, I know then the frequency of the recessive allele is 11%. And as you can imagine, 89% plus 11% gives us 100%, which is the total frequency of both alleles. Remember, the frequency of P plus Q has to be equal to 1. The second way of calculating the frequency of those alleles is from the genotype frequency. How do we do that? First of all, I have to look at the number of individuals for each of the genotypes. So the genotypes here are homozygous dominant, heterozygous, and homozygous recessive. I told you that I have 79 that are homozygous dominant, I have 20 that are homozygous recessive, heterozygous, and I have only one that is homozygous recessive. Now, knowing that, I can calculate the genotype frequency, meaning that I divide the number of individuals in that category by the total number of individuals. Therefore, 79 divided by 100 equals to 79%, or 0.79, which is also 79%. And you can see that we can calculate the genotype frequency of the other for the other genotypes. Knowing the genotype frequency, I can now calculate the allele frequencies. How do I do that? Well, for the allele A, how do we calculate that? The allele A is present in the 0.79, correct? And is present half in this 0.20. So this should be 0.1, which equals to 0.89, which is 89%. How about the allele recessive? Well, I know that Some of them, or actually half of them, are here in the heterozygous, so I'm going to represent that by 0.1, and the other left over is right there, so I'm going to represent it by 0.01. So that is 0.11, which equals to 11%. So you can calculate the frequency of the alleles either by counting the number of alleles or by the genotype frequency. In the exam, I might give you one or the other for you to calculate those frequencies. Okay? If I give you the genotype, then you can calculate the frequencies using those two, either of those two methods. The question is now, how do I calculate the frequency, or how do I make things a little bit more complicated when we add more than one allele, as we saw the case for multiple alleles, I believe in either class 3 or 4, the blood type, right? The blood type has the following alleles: IA, IB, and lowercase I. Here, of course, is not a case of just dominant versus recessive alleles. So how do we solve the situation where we have 3 alleles? Simply by adding one more letter to that equation, okay? So here we have, let's just say that instead of saying, instead of working with the blood type, because that's going to be my example, let's say that besides of having A, capital A and lowercase a, I have another allele here called B. The sum of their individual frequencies have to equal to 100%, or 1. Now, our equation that was initially given, so for 2 alleles, our equation was p squared plus 2pq plus q squared equals to 1. Now the equation for 3 alleles becomes a little bit more complicated because we need to add this other guy in the game. Okay? I am not explaining how we get to these equations because this is mathematical assumptions, right? So this is simply doing something like this, and then you get to this equation. So I'm just skipping that step and just going straight forward to the to what is interesting here for our class. Like I said, my example here will be for the blood type based on the system ABO. As you might remember, this is a type of multiple alleles, right? Because here we have these 3 alleles and these blood types. Type A can be either due to the IA or IA lowercase I. Type B can be IB or IB lowercase I. Type AB can only be due to IB, and type O can be only due to homozygous recessive. Knowing that, how do we apply this Hardy-Weinberg equilibrium to this scenario? Here, I am already giving you the frequency of the alleles. I'm telling you that the frequency of the allele IA is 38%, the frequency of the allele IB is 11%, and the frequency of the allele lowercase I is 51%. Knowing that, I'm asking you to calculate, or the table is showing you how to calculate the genotype frequency for each one of these genotypes. Okay? So, for the genotype where we have both homozygous, this is how we calculate. For when we have one homozygous, when we have the heterozygous, this is how we calculate, and then so on and so forth. Okay? The phenotype, of course, will be different because these two guys can be the same phenotype. Therefore, the phenotype frequency is just the sum of these two genotype frequencies. Okay? For the B is the same thing. You have these two genotypes that can give you the type B. Therefore, the phenotype frequency for B is just the sum of the two genotype frequencies. All right? For the AB, this is what we get, and for O, this is what we get. So here I am just showing you how to calculate those frequencies when we have multiple alleles. Okay? But it's very simple. We just added another factor, another letter to our equation. The knowledge about the Hardy-Weinberg law also help us to understand or to calculate the frequency of the heterozygous in a population. Remember that I told you that most of the genes that we have for many traits that we have, they happen in a population in heterozygosis, meaning that most of the individuals will be dominant in one allele and recessive for the other allele. Especially when we look at some disease, right? I don't want to use the vocabulary most, but there is a great number of diseases that are autosomal recessive, meaning that when we have this genotype, a homozygous recessive, those diseases are manifested. Therefore, this type of genotype is actually selected, and over generations, the frequency of this genotype is reduced. But here, I want to calculate the frequency of the heterozygosis in a population, and we are going to use the example for cystic fibrosis. Okay? So our population, we're going to use a Northern European ancestry. So here, I am telling you that of this population, the incidence of cystic fibrosis is 1 in every 2,500 people. Okay? So in a population of over 2,000 people, one person will be positive, will have the cystic fibrosis. The cystic fibrosis is an autosomal recessive trait, meaning that we would need the genotype to be both recessive for that person to be to manifest the cystic fibrosis. What I want to know here is how many of those individuals are just carriers of the condition. Okay? They don't manifest the mutation, they just carry the mutation. So, Knowing the incidence, how many of those individuals in that population are or they are positive for this autosomal recessive trait, we can calculate the frequency of the heterozygosis in this population. Okay, knowing that the incidence is 1 over 2,500, I know that 4% of this population has this autosomal recessive trait. Okay? Knowing that this autosomal recessive trait equals to 4%, I know that this 4% is my Q squared, right? Because remember, A dominant is represented by P and A recessive we are representing by Q. So 4% is actually Q squared. 4% can also be represented as 0.04. Well, I can do some math here and actually find the value of Q, which is the first step that we need to take in order to understand how many of these individuals are carriers. Okay, how do I solve this? By applying math laws. So I isolate Q, now I calculate the square root of this value, and my Q should be what? 0.2, right? So I know that the frequency of my allele A is equal to 0.2 or 20%. Remember that we sum these frequencies, right? We sum the frequency of the dominant allele plus the frequency of the recessive allele, and we expect that to be equal to 100% because that's the total number of alleles in that population. Well, I can also write this equation as this: P plus Q equals to 1 because P is the frequency of the dominant allele, Q is the frequency of the recessive allele, and we know that this should be equal to 1. If I know how much my Q is, I can isolate P to find out what is the frequency of my dominant allele. In order to do that, I just replace the value of Q in my equation, and as you can see, P should be equal to 0.8 or 80%. So the frequency of our dominant allele is 80%, which is the second step now solved. So we solved 1 and 2. The third step now is very easy, right? Because to calculate the frequency of the heterozygosis in this population, we can just use the Hardy-Weinberg law. So we can apply this to actually calculate the frequency of our heterozygous, or let me represent this by the frequency of our heterozygous. Okay, so this should be 2 times 0.8 times 0.2, and this should be equal to 0.32 or 32%. So in that population, in this total number of individuals, which I told you is 2,500, I know that 32% of them will be or will have the genotype heterozygous like this. Knowing all these values, I can also calculate, and I will leave it up to you, the frequency of how many will be homozygous dominant and how many will be homozygous recessive. We have all these values and we are able to calculate all of them. Okay? We can actually represent the Hardy-Weinberg law, or this model, by plotting these equations or plotting our understanding about these values in a graph. So this graph here shows the relationship between the genotype and the allele frequencies, because that's what we are interested in understanding when we studied this model. Okay? And by genotype, remember, we were interested in the homozygous dominant, heterozygous, and homozygous recessive. The frequency of the homozygous dominant was given by p squared. The frequency of the heterozygous was given by 2 times p times q. And the frequency of the homozygous recessive was given by q squared. Okay? Here, this graph is showing you the relationship between the genotype frequency and the frequency of the alleles dominant and recessive in that population. So let me just erase this so we can start from the beginning right here. Okay, so when we look at this graph, this is what we have to understand. On the bottom of the x-axis, we have the frequency of this allele. And you see that from the left to the right, the frequency is increasing, which is the opposite for the frequency of the recessive allele. From the left to the right, the frequency decreases, and from the right to the left, the frequency increases. Okay? The genotype frequency is exemplified or is plotted on the y-axis. This is how we interpret this graph. We know by looking at this graph that if the frequency of the allele A dominant, so if the frequency of the dominant allele increases in that population, this will cause, this will cause an increase in the frequency of our homozygous dominant genotype. Okay? So that is represented in the green line, as you can see right here. So the green line is showing you that if the frequency of the dominant allele increases in that population, then that leads to an increase in the frequency of our homozygous dominant genotype. The opposite is also true. Looking at the red line, we can observe that if the frequency of the recessive allele increases in that population, what happens is that there is an increase in our homozygous recessive genotype. Okay? So here we're talking about the allele, and this is the genotype. type, allele, genotype. Because that's what we are trying to understand with this model. What is the relationship between the allele and the genotype? So in a population with those conditions that we observed, that we with those assumptions that we had at the beginning, this is what's going to happen if the frequency of the alleles change, the frequency of the genotypes also change. As you can see, there is a point of equilibrium, which is right there. In that point, the frequency of the dominant allele is the same as the frequency of the recessive allele. In that case, the genotype that will be more expressed, or that will be more present in that population, will be our heterozygous, okay? Which again is represented by 2pq. All right, so in that point of equilibrium where the frequency of both alleles is the same, then we have a more prevalence, or a higher prevalence, for the heterozygous genotype. Okay? When we studied the Hardy-Weinberg model, one of the assumptions, or generally we were assuming that this population that we were studying, that we were observing the allele frequencies, therefore the genotype frequencies, these populations, they were stable, right? There was no migration, there was random mating, and all of those assumptions that we had. However, when we look at the natural world, that is not always the case, or very, very often, almost 100% of the populations are not stable and fixed, right? Because populations go through changes, and those changes occur in the level of the alleles, therefore of the genotypes. The frequency of those alleles change in a population over time. And that change in the frequency of the alleles is what we understand by evolution. So evolution actually, very simply, means that the population over time is changing the frequency of the alleles. And we can observe that by observing the genotype and the phenotype in that population. Okay? Evolution is changing. That change doesn't necessarily mean that we're changing for a better organism. Okay? The change in those alleles can cause those individuals to be less fitted for that environment. Therefore, they won't be selected for that environment, right? So not always evolution is causing a change not always the change in the allele frequency is good, okay? It can cause deleterious changes, and sometimes those deleterious changes, they do not they don't prolong for too long in that population. The changes that can happen in the allele frequencies within these populations can occur by 4 different mechanisms, and we are going to study each one of them. These mechanisms are mutation, genetic drift, migration, and natural selection. This slide is a summary of those 4 mechanisms. What we are going to do from now on is to study each one of these mechanisms. Remember, all of these mechanisms will cause changes in allele frequencies, okay? Because we have more than one allele in that population. All right, so the first one we're going to start with is the natural selection. And of course, you probably heard that natural selection was a theory postulated by Darwin, correct? And I already talked about this a little bit at the beginning of the class. However, I also think it is important that we give some credit to other people that came before Darwin, and those ideas developed by those people somehow influenced what Darwin proposed and how he formulated for the natural selection theory, okay? Because we've got to understand that science gradually changes, right? It is not that one beautiful day Darwin woke up and suddenly thought, oh my goodness, yes, natural selection explains evolution. No, those changes in the scientific beliefs, they happen gradually in very small steps, baby steps, okay? What were those ideas that somehow shaped Darwin's natural selection theory? First of all, one of the important ideas was that the Earth was a very old planet. Okay, so that idea was already postulated by some scientists due to their study of the soil, of the rocks, and all of this material, right, present in Earth. Not only that, but also some other scientists were already finding out that species go to extinction. They no longer exist, right? And that was observed due to the presence of fossils that were discovered by the scientists that you are probably reading the name right now. So this guy actually observed or found out those fossils, and he couldn't actually match those remainings with existing species in that by that time. So, one of the conclusions is that those species, they die, so they suffer extinctions. One important idea as well was that those species, they change. And one of the important names that suggested that species change throughout time was Lamarck. So Lamarck was one of the guys that postulated that species change from one generation to the other. So parents pass their genetic information to their offspring, and the offspring is somehow different from the parents, okay? But here, the idea behind Lamarck's theory for those changes in the species were somehow different from what Darwin proposed. Lamarck thought that those changes were due to the use or not use of the of those traits. Okay, so for instance, this is one very classical example that you probably heard in school before, but it was not proposed by Lamarck at all. Okay, this is just an example that we used to simplify what he was trying to, what he came up. Okay? So the idea here is that if in a field there are lots of trees available, okay? And some of those trees, they have a very good foliage. They have a very good leaves available at the very top of those trees. Therefore, some animals like giraffes would develop a long neck to reach those better leaves at the top of those trees. And they would somehow push their neck up and up to the point where they would reach those better leaves at the top of the tree. So for instance, the individual number 1 would have a few inches higher neck. Then individual number 2 would have more or higher, even higher neck, even longer, neck. Then individual number 3 would have an even longer neck, just by pushing that neck over and over. Okay? So if the individual was using that trait or that anatomical part of the body, then that trait would evolve or would be expressed in that population. But if the individual was not using that, then they would lose those traits one generation after the other. Okay? Also, one of the ideas that influenced Darwin's thinking was that was about the Earth's features and how those things change. And that was based mostly in the geological observation of the Earth. Okay? And those ideas together kind of culminated or helped Darwin to develop this idea of natural selection, which is what we are going to look in the next slide. We will now start studying about natural selection. The natural selection is one of the driving forces for changing the allele frequency in a population. Always remember that when we say allele frequencies changing in that population, we refer that as evolution. So, evolution means changes in allele frequencies. Okay? And one of the mechanisms that promote changes in the allele frequencies is natural selection. In the following slides, we are gonna develop this idea about natural selection. So we are gonna build up the knowledge about natural selection. But at the bottom of this slide, you already have a very summarized understanding of what natural selection is. This will be true for the other mechanisms as well. Before I introduce the mechanism, you will see this statement in red at the bottom of my slide, which means that this is a general summary of what that mechanism actually means. So I do recommend that you pause for a moment now, this video, to read this statement. Okay? Because again, it's just a summary of what natural selection is. So natural selection is actually a mechanism, a theory for how evolution occurs. And even though we give a lot of emphasis in the work developed by Darwin, there is also another researcher named Alfred Russel Wallace that also proposed some ideas about natural selection, but independently of Charles Darwin. Okay? Even though we only, for some reason, we only discuss about Darwin's discovery usually in class. Also, other researchers contributed to the development of this idea about natural selection. The main thing here that we need to understand is that within a population, the individuals, they exhibit variations in their phenotype. Meaning that when you look at a population, the organisms, the individuals of that population, they don't look the same. So the traits, they vary in this population. And that variation in the traits, in the phenotype, is passed from one generation to the other. So we say that those variations are heritable. Okay? Meaning that from one generation, it is passed on to the other generation. Some of the phenotypes in that environment where the population is located, they are more successful in surviving at the conditions of that environment. Okay? So if you think of any place, that environment is submitted to certain natural conditions. That can be dry seasons, wet seasons, fires, natural fires, wildfires, or any other natural event. Some individuals are more successful in surviving to those events, to those natural events. Those individuals that are more successful in surviving to those natural events, they tend to leave more offspring than those who died, right? If those organisms that are not as successful at surviving to those phenomenon, they ended up dying. Therefore, the offspring of these organisms is lower in number. And it gets lower and lower every generation. Those that are more successful, they have more chances of reproducing. Therefore, their offspring is more numerous over and over. Okay? Because of that, we call those phenomenons that are happening within that environment of pressure, of natural pressures. Okay? So that environment is putting pressure onto those individuals. And some of them are better fitted to that environment. Okay? So let's think that you have a population of a certain organism. And within that environment, you have two types of individuals in that population. You have one that has a very thick skin and one that has a more thinner skin. Let's say that this environment is submitted to constant fires. And let's say that over time, those that have a thicker skin are protected from the fire. Those that have a thinner skin are less protected from the fire. Over time, you will see that those that have a thicker skin will be more numerous in that population. Therefore, they will produce more and more offspring with that trait. Over time, the number of those individuals with a thinner skin tend to be reduced to the point where this organism no longer, after many generations, will no longer exist. And only the one with a thicker skin now exists. Does that make sense to you? And if it doesn't, then you stop and try to listen to this audio again. What I am saying is that fire in that environment is a selective pressure, is a natural pressure that ended up selecting in a natural way which of these two organisms will survive. The organism who survives leave offspring. That trait, the thick skin, is inherited from one generation to the other. That is the base of how natural selection occurs. Is selection because one type is selected over the other. It is natural because it is the environment due to the pressures that ended up executing that selection. Okay? There is no human influence, let's put that way, in selecting those traits. In order for natural selection to take place in that environment, there are three conditions that need to be met. Okay? The first one is the variation of the trait that we are analyzing. In my example, the trait that I gave you was the thickness of the skin. But that the trait can be whatever that confers to that species or to those individuals a better chance of surviving to those selective pressures. Okay? So the first thing is that there should be some variation of the trait. Because if all the organisms look the same, then when there is a selective pressure, all of them might die. For instance, in my example, if all the organisms in that population only have a thinner skin, then when the white fire comes, what happens is that they all die, and that species come to an extinction. The different versions of that trait is due to the alleles, right? Remember, the genotype confers or helps shaping the phenotype, which is the thing that we look, which is the visual appearance, the visual manifestation of the genotype. However, what's guiding the genotype are the alleles present in the chromosome. Okay? And here in this example, in each chromosome, I am representing a different version of that allele. Remember when we discussed that the gene that encodes for that trait can come in different flavors. This is the main idea here. There should be variation in that trait, meaning that the gene that codes for that trait comes in different flavors. The second aspect of the natural selection is that the trait has to be heritable. So the trait must pass from the parents to the offspring. So the second condition here is heritability. So first is the variation of the trait. The second is heritability. So that trait has to be passed from one generation to the other. The last condition here is that there should be a differential reproductive success of those individuals. Let's say that the trait that we are analyzing, which was the thickness of the skin, does not actually influence the reproduction of those individuals. If that trait does not influence the reproduction of that individual, then that trait will not be submitted to selective pressures. Okay? However, if that trait does contribute to the reproduction of that species somehow, then that trait will be submitted to those selective pressures. Okay? And one trait to be selected over and over in that environment has to give a better ability for that species to reproduce. In the case of the skin, the thickness of the skin is the more number of individuals that survive that have a thicker skin, the higher the chance of then mating and producing more and more individuals with thicker skin. Okay? So, these individuals that we are analyzing for that version of the trait, they have to be suited for that environment. And being suited, they leave a more numerous offspring in that environment with that trait. So let's look at my example here. So I told you that the trait that we are looking has some variation, right? And I am representing the variation with different colors of those alleles. As you can see here, this allele after the selective pressure only left one individual surviving that environment. But this other allele, look, left more individuals. Therefore, this allele will be positively selected in that population. And this other allele might not exist after some generations. Okay? Sometimes those alleles can exist as a recessive allele, for instance, and is passed in heterozygosis, right? In a condition like this. And we've talked about this before. As a consequence of the process of natural selection, what happens in terms of population and as well species is change. So again, natural selection is a mechanism that drives changes in the allele frequencies in that population. So here, the traits that confer to those individuals a reproductive advantage will increase in frequency. not only the traits, but also the alleles associated with that trait. So, as the trait increase in frequency, so will the alleles increase their frequency. Because those traits will cause the population to be better adapted to that environment. Over time, if the differences, if the variation in that original population becomes greater and greater between individuals, we can actually have a new species appearing in that population. So let's say that at the beginning, you had a population of individuals. And here I'm just going to represent with dots. Okay? Let's say that over time, you also had here some individuals that had a different trait. And I am representing them with the blue color. Let's say that over time, in this population, the ones with the red color ended up dying for some reason, leaving less and less individuals or offspring. But the ones with the blue color here, as you can see, is producing more and more offspring over time. There might be a point in the history of these individuals that what will happen is that they become reproductively isolated, meaning that the individuals here that were red and the individuals that were blue, they no longer can mate. So what was one single species here now becomes two species in the future. Okay? Because the changes in the frequency of those alleles became so great that it created two distinct populations, two distinct species. Okay? And with that, they become reproductively isolated. They no longer can mate and produce a viable offspring. This is a concept that will come over and over throughout this class, because this appearance of a new species can also be driven by the other processes that can drive evolution. Okay, so mutation can also cause the appearance of new species. Genetic drift can also cause the appearance of new species, and other evolutionary mechanisms can cause the appearance of new species. So this has to be well understood from now, right now, because this will come up in the future slides. Here I am just giving you a different example of how natural selection works in nature. So here we are looking at a population of rabbits. And within that population of rabbits, you have different individuals that have different speed. Some individuals are faster, some individuals are slower. There is a degree of variation in that trait. And that might be because some of them have more muscles in their legs. Some of them are just lighter in their weight. That can be, the speed can be do anything. But the speed is a trait that we are looking here, is a feature that we are looking here in this population. This, whatever that's causing these animals to be running faster or slower, is passed on to the next generation, from the parents to their offspring. For instance, if it is more muscle or less muscle in their legs, then there is a gene that codes for more protein or less protein in their legs. So that gene is inherited from one generation to the other. But here in this environment, we are going to now add a predator, which is a fox, that will eat those rabbits. Here, as you can see, the rabbits, and probably I should be pointing with the laser here so you can also see what I'm saying. Here, those rabbits that don't run as fast like these rabbits, they ended up being predated. They ended up being eaten by the fox. Meaning that these guys are better adapted, better fitted to that environment. So the fox represents here a selective pressure. These guys are better adapted to that environment because they can run faster and they escape from the fox. These other ones, they don't run as fast, they are slower, therefore they are predated by the fox. And what happens here over time, as you can imagine, is that these individuals over time will leave a lower number of offspring, and these will happen to be more numerous because they are not predated much by the fox, and because of that they leave more and more individuals. They leave a more numerous offspring every generation. So this trait is selected to continue in that environment, and this trait is selected to no longer exist in that environment. Okay? So here is just another example where the fox is the selective pressure, the trait is the speed, and here we are looking how the green rabbits, because they run faster, therefore they are more numerous, they can leave more individuals in the next generation. They can produce more individuals for the next generation. Therefore, this trait conferred to this species a reproductive success because they can reproduce more easily than these can, because they can find more mates than these guys can. As I mentioned it before, there are some alleles that can be fatal to a certain population. And usually those alleles that are fatal, for them to be fatal, they've got to happen in a homozygosis scenario, meaning that both alleles have to be equal. In some of the examples that we used in previous classes, and in the example I will be using in the next slide, let's assume that the allele recessive is lethal. okay? And for this allele to be lethal, it has to happen in homozygosis, meaning that both alleles have to be recessive, like this, okay? Because this is a lethal allele, what will happen here is that the natural selection will work in a way that these individuals will over time be less numerous in that population. And that happens because if this is a lethal allele and the individuals are homozygous, then these individuals ended up dying without leaving a offspring. If they don't have kids, if they don't have an offspring, then the frequency of that allele starts decreasing generation after generation. So in the next figure, we will see how this the frequency of this lethal allele changes according from one generation to the other. So this graph is actually summarizing how the frequency of the allele A changes over generation. If this allele A is a lethal allele, then the environment will select against that lethal allele, because those individuals, they do not reproduce, right? So that trait is not giving them any reproductive success. Here, what we can see is that as generations go on, the frequency of the recessive allele in that population is decreasing, okay? And what's happening with the frequency of the dominant allele is increasing generation after the generation. So you see here that in generation 1 was 67%, and then now after 100 generations, the percentage of this allele increased by almost 100%. Because the frequency of the alleles are changing one generation after the other, we also observe changes in the frequency of the genotypes, as you can see here, demonstrated by p squared, 2pq, and q squared. You can see that the frequency of the genotype homozygous recessive is decreasing, and that is because the frequency of the allele is decreasing from one generation to the other. And that is because, again, this allele is deleterious, is lethal, meaning that the selective pressures will try to get rid of this allele, because these individuals do not leave a offspring, so they can't reproduce. So there is no reproductive success associated with this allele, neither with this phenotype. However, there are different degrees of selection against those alleles. Usually, if the allele is lethal, the selection will be really strong against that allele. But if this recessive allele is not lethal or is a non-lethal allele, the selection against this allele can vary from a very strong selection to a very weak selection. In this graph, we are representing this concept. Again, I am showing you the frequency of the recessive allele in that population. In the x-axis, you can see the number of generations over time. You can see that In the red line here, if the selection against that allele is really strong, then the frequency of that allele over time decreases, which also decreases the frequency of the genotype. However, in the other extreme, and let me erase everything here. In the other extreme, if the selection against this allele, this recessive allele, is weak, then what we observe is that the number of, the frequency of the genotype doesn't change even after many generations, or change in a very slow manner, okay? So here, this graph is showing you that the selection against that recessive allele can be strong, which decreases very drastically the frequency of that allele, or very weak, which causes change in the allele frequencies, but very slowly over those generations. Those are the two main conclusions that we can take from this graph. The first one is that even when the selection is weak, we still have changes in the allele frequencies and in the genotype frequencies, but in a more slower pace. If that selection is stronger, of course, the changes in allele frequencies will be much faster. Also, we can observe differences in the fitness among the genotypes. They must be large for the selection to produce rapid changes. Okay? Remember, there should be differences in the reproduction, in the reproductive success for that genotype to be selected in that environment. Several types of natural selection can be observed in a population. Here, the three types that we are going to study in the upcoming slides are the directional selection, stabilizing selection, and disruptive selection. To make things easier to understand, let's consider that in this next few populations that we are going to use as an example, we have three phenotypes. We have our homozygous dominant, our heterozygous, and our homozygous recessive. Each one of these genotypes give one certain phenotype. So here we have phenotype number 1, phenotype number 2, and phenotype number 3. The name of the selection suggests us what the selection is doing to these phenotypes. And we are going to look at them very carefully, and I'm going to give an example. But in a directional selection, either this phenotype or this phenotype will be selected. Okay? So selection will favor one and will be against the other. So here we can use the comparison this or this. In a stabilizing selection, it will be the intermediate phenotype that will be favored in that location. Here, the selection will be against the extremes. So here, the selection will act against these two phenotypes. So in a stabilizing selection, this phenotype will be more prevalent in that population. In a disruptive selection, as you can imagine, now between those genotypes and phenotypes, the selection will tend to decrease the number of individuals in the intermediate phenotype. And those extremes will be favored, okay, together. So here we can use the and to help us, understand. Okay, so while in the directional selection we have either phenotype 1 or 3 being selected, in the disruptive you have both of them being selected at the same time. In this disruptive selection, what can happen sometimes is that these individuals will become different populations because they become so different over time, one generation after the other, that we ended up isolating those individuals reproductively. So now let's look at each type of natural selection. Like I said, in a directional selection, the traits at one end of the spectrum of those phenotypes become selected for or against. So if you think that in that spectrum you have phenotype 1, phenotype 2, phenotype 3, either this or this will be selected, okay? And which one will be selected in favor will be more prevalent in that population and leave more offspring. Here's an example of this directional selection. Let's look at the average beak size in the finches, in the birds that Darwin studied when he went to the Galapagos Island. There was an observation here with these birds that in dry years or dry seasons, the average beak size of these animals would increase. And they observed that during wet seasons or wet years, the average beak size would decrease. Meaning that in dry years, the phenotypes with bigger beaks would be selected, and during wet years, the phenotype with smaller beaks would be selected. So you can see that no intermediate was selected here. There is no medium-sized beaks here. It's either a big or a small beak that is being selected according to the changes in the environment. And why is that the case? Well, that happens because in dry years, those birds, they have to eat a large and hard seed. Therefore, because it is harder to break that seed and the seed is larger, then it is better for that individuals to have a bigger beak. Therefore, those that have bigger beaks, they have more access to food, they survive, and they can reproduce more often or can leave more offspring than those ones that have a smaller beak. However, in wet years or wet seasons, the seeds now, they became small and soft. Therefore, you don't need actually a bigger beak to break those seeds. A small beak here now works just fine to access that food. Therefore, this trait is favored, and the other trait is selected against. So here in directional selection, either one of those extremes are selected, okay? Not both of them at the same time. That doesn't mean that during dry seasons you don't have animals with smaller beaks. Yes, they are still there, but in a lesser in a they are less numerous in that population. Okay? Same thing in the wet seasons. That doesn't mean that the ones with bigger beaks are not present there, but they are less numerous in that population. And that causes fluctuations in the frequency of the alleles and in the frequency of the genotypes. The second example that we have here is the stabilizing selection. In the stabilizing selection now, it is the intermediate phenotypes that will be favored. Now the extremes are selected against. So if you think about our initial example, the extremes here will be selected against, and the middle one, the intermediate, will be favored. Okay? Here, the consequence of this type of selection is that the population, the variance, the variability in this population reduces, right? Because now that intermediate phenotype is being favored. So the variability usually decreases. An example here is the birth weight in humans. What happens here, according to this graph, is that if the kid, by the time that they are born and delivered, if they are too small, or if they are not as heavy in terms of body mass, they ended up dying more often. However, if the kids are fatter, then they also ended up dying. The births here are not successful because now the delivery is much more difficult. Therefore, what's going to be selected are those kids that have an average weight. By having this average weight, those kids, or that trait, is selected by that selective pressure, and that is the trait that will be present in the population. Okay? So being too small or being too overweight here in this case does not confer a reproductive success to that population. But being or having an average weight gives a higher reproductive success to that population. Therefore, this trait in the middle will be selected in favor. Okay, and the other ones in the extreme will be selected against. The last case of natural selection is the disruptive selection. Here, the intermediate phenotype now will no longer be selected or will be selected against. Here, both of the phenotypes in the extremes will be selected in that environment. If we go back to our example, we have this one being selected against, and this one being favored, and this one being favored as well. Okay? Here, what we are going to have with the variation in that population is a type of bimodal distribution for that trait. Because as you can see, this is very distinct of this. They are completely different. What happens is that over time, this disruption can actually lead to the generation of a new species. And then these two types of species, they are not compatible anymore, meaning that they cannot reproduce anymore. Here's an example with flies, okay? With the bristle number in flies. What you are observing here is that in generation 1, there was a mix of traits in the number of bristles. You had flies that had very little number of bristles to very high number of bristles, and the ones that had a intermediate number of bristles. However, as the generation goes on, those traits in the extremes, they start being selected. And after 12 generations, as you can see, you selected those that have smaller bristles and those that have really large, not smaller bristles, sorry, a lower number of bristles, and those that have a higher number of bristles as well. The intermediate phenotype now no longer exist or exist less number of those individuals. Here, as you can see, those two populations now, they do not overlap anymore. And after 12 generations, it is possible that here we created now two different species, okay? Because probably they no longer can reproduce. That's the effect of the disruptive selection. Putting all together, we see that a directional selection, the population will shift to one extreme or the other of that phenotype. In the stabilizing, here you're gonna favor those intermediate phenotype, which ended up to reduce the diversity in that population. If everybody looks the same, then diversity is lower in that population. The disruptive selection, here you're gonna select two phenotypes at the same time. And here you're gonna have two distinct populations from this bidirectional selection. Those three types of selections can be represented graphically like this. So here you have the directional selection where one trait is favored against the other. Here you have the stabilizing selection where the intermediate trait is favored. And here you have the disruptive selection where over time that trait in between is selected against, and what's favored is the extremes. Just so you know, if you try to read those graphs, 0 here represents your initial population, and S represents your population after a certain amount of time. Okay? So go over these graphs and try to correlate each type of natural selection. Another biological phenomenon or another mechanism that can help creating or changing the frequency of the alleles and the frequency of the genotypes is mutation. For the next two slides, or in the next two slides, we will see how mutation works and how we can identify a mutation in a population. But here, at the bottom of my slide, you can already see a big summary of what we will work through this concept. What I want to highlight about this concept, about this mutation as a mechanism for evolution, is that mutation is the only way that new alleles can be created. When we talked, for instance, about natural selection, one of the things that I hope it became clear with you, for you, is that natural selection does not create a new allele. Natural selection selects the alleles available in that population already, okay? Favoring either the extremes, favoring the middles, or favoring in one side or the other, right? Within those three categories of natural selection. There is no new allele forming in the case of the natural selection. That's also the case for migration and genetic drift, which are the other two mechanisms that create or change the allele frequencies, therefore are evolutionary mechanisms, okay? So of the four mechanisms that we are discussing in this class, mutation is the only one that actually creates a new allele in that population. As we know, the DNA is the genetic, or carries the genetic information of a certain species. The DNA is composed of nitrogenous bases, right? And we represent that DNA by a double strand molecule, okay? Which we also studied in the first class, that this forms a double helix molecule. In that molecule, we have the bases, right? So the bases, they make up sort of the steps of this ladder molecule, okay? And this portion is composed of the sugar and the phosphate. Here, on the steps of that ladder, we have the bases, and that can be A, T, C, G, another whatever, A, okay? The sequence that we have here is what we call gene, and that gene codes for a protein. On your left, you are seeing an example of a normal sequence of DNA that produces a normal protein, and that causes the creation or the generation of a normal phenotype in cats. However, due to a mutation, which here we are going to understand mutation as a replacement on the base pair of that sequence. So you can see that, I'm going to erase the ink here so you can have that picture. You can see here that in the sequence, only one base pair is different compared to the normal sequence. This new sequence we call the mutated base pair sequence. This mutated sequence will then code for a mutated protein, which then gives rise to a mutated phenotype. In this case, this phenotype is given by cats that have curled ears, okay? So mutation within a population create new alleles. And by creating new alleles, because it switches the base pair in that sequence, that will cause changes in the allele frequencies. Here, to measure or to observe the actual mutation in a population and to actually observe those changes in the allele frequency, this mutation must produce a allele, And that allele produced by a mutation has to produce a distinct phenotype, a phenotype that is different from one to another. Okay? So here you can see that this phenotype is completely different than this phenotype. So we can certainly identify that a mutation happened that created this second mutated phenotype. Besides of the fact that new mutation must produce a distinct phenotype, the traits that we are observing, they either must be fully expressed, so we must see the phenotype, or they must be completely penetrant, meaning that even though we don't see the phenotype, we can at least detect in the genotype. And we detect that in the genotype by analyzing the DNA via PCR. And we talked about this very briefly when I showed you some electrophoresis gels. The fact that the trait must be fully expressed or completely penetrant in that population is because we need to identify those mutants, either by observing with naked eyes or by knowing or by having ways to know their genetic composition. One important thing here is that identical phenotype must never be produced. Otherwise, you can't distinguish whether you have a mutation or not in that population. We sometimes have a misconception about mutation because you probably watched a lot of superheroes cartoons like X-Men. Here we must understand that the mutations, they not always will actually provoke a drastic change in a population. Not all mutations will lead to a gain of function in that trait or a loss of function in that trait. Some mutations are actually neutral and they don't result in any identifiable phenotype. We can perhaps identify that the mutation is there doing genomic sequencing, gene sequencing, but probably that does not actually change the protein to a level that we would see in the phenotype. So not all mutations will lead to those gain of function or loss of function mutations, okay? I am sure that if we get our DNA and if we sequence our DNA, we will find out that for some of the genes that we share, we will see those base pairs slightly different, but we'll be expressing the traits in the same way. Okay? So not always the mutation will lead to a drastic change in that population. Another mechanism that can change the allele frequencies in a population causing evolution is migration. Migration here can be understood as the gene flow between populations. This is the literal physical movement of those species from one area to the other. And that creates the gene flow, the gene moving from one area to the other area. Again, at the bottom of this slide, you find a big summary of what this migration means in the context of evolution. Here, I am saying that migration, which can also be understood as gene flow, occurs when those individuals in that population, they physically move between those two populations. Okay? There are two consequences from that migration. The first one is that it can reduce the genetic difference between two populations because you can dilute the genes between those two populations, or it can increase the genetic variation. So both outcomes are possible from this migration. Remember that migration is one of the things that we do not count when we are talking about the Hardy-Weinberg law. Okay? Remember that in our ideal population, when we studied this model, we assumed that migration did not happen. Here on the right, you can see an example of how migration can change the allele frequencies in two populations. So here you are looking at two populations, population number 1, population number 2. Those two populations, they are separated by a mountain range. In population number 1, you have individuals with, for instance, alleles. Let's say that these are dominant, homozygous dominant, and let's say that these are heterozygous for this gene. The population 2 is part of the same species as population 1. So here they also have alleles homozygous dominant, but as you can see, only a small fraction of them are heterozygous. What happens now is that when a portion of this group of individuals start moving, migrating over the mountain range, now they bring their genetic load to this second population. And after the migrating event happens, you can see that now you have a greater amount of individuals that are heterozygous in that population, and here you have a reduction in the number of those individuals. You are literally carrying the genes from one population to the other population. Is literally the physical movement, the physical flow of those genes from one side to the other side of the mountain range. Okay? There are some cases in nature, and we are not going to talk about those examples because the example I am bringing here is completely different, is more related to humans. But there are some examples where population 1 stayed in the same place, and let me delete all this so I can discuss a little bit better. There are some examples in nature where population 1 stayed where they are, and this portion of population, for instance, was or migrated to a completely new space. Here, let's consider that no population was existing here before. These new individuals, they become a new population here. And these new individuals, if they are apart from population number 1 for successive generations, there is a point in their history, in their natural history, that they become reproductively isolated from each other, meaning that they are no longer from the same species. Now you have two distinct populations with two distinct species. Okay? Big migrations of the animals across the globe helped creating different populations depending on where those animals resided. Okay? There are tons of examples, but due to the timing, we're gonna try to keep it more straightforward with the example of the human population. Here you can see the migration of individuals from Asia into Europe. These individuals here in Asia, they carried with them a lot of the IB allele in their blood, in their DNA. This, as you can see, this allele is highly prevalent here in Asia. And as these people migrated into Europe after the fall of the Roman Empire, they carried that gene, that genetic information with them, spreading that allele throughout Europe. Okay? But as you can see, there is a gradient of the distribution of this allele between Asia and Europe. And that is because of the migration that happened right after the fall of the Roman Empire. Another mechanism associated with changes in the allele frequency in populations is the genetic drift. Genetic drift literally means change, right? So this means that the genetic pool, the gene pool in that population is changing towards one direction. Okay? Here, one important thing to observe in the genetic drift as a cause for evolution is that the alleles that we are studying or that we are considering in that population, they do not influence the reproductive success. Different from natural selection, where the selection of that individual is related to whether those individuals can produce more or less offspring in that population. Here, the alleles do not influence the reproductive success of that population. Okay? Again, at the bottom of the slide, you can see a summary of what we understand by genetic drift. In general terms, the genetic drift means random fluctuations in the allele frequencies. The genetic drift usually happens in small populations. This can happen just by chance alone, but we are gonna see two phenomena that can happen in a population that are associated, that can cause this genetic drift. The first one is the founder effect, and the second one is the genetic bottleneck. In general, this picture on the right is summarizing what the genetic drift causes in a population. At the beginning, right here, considering that we are studying a population of cats. In this population, let's consider that we have some cats with white fur and we have some cats with orange fur. Okay, as you can see right here in their faces. As you can see, the one with some white fur is dominant in the population compared to the one that has all orange fur. Here, the trait fur being white or orange does not actually influence the reproduction of these cats. Okay? Doesn't matter which color you have the same chance of mating, the same chance of finding a reproductive partner in that population. But here, what happens during the genetic drift is that you have more individuals now with the orange color and less individuals with the white color. So you can see that the gene pool is drifting, is changing towards one direction. Okay? Not because these cats have a higher chance to reproduce. It is totally through a random process. Okay? A random fluctuation in the allele frequencies. What can happen throughout the time is the fixation of one single allele in that population. So here, as you can observe in my schematic representation at the bottom, you have this population with two types of fur, let's say, two types of alleles. But for some random effect, that can be the founder effect or the genetic bottleneck, here now this allele starts to become more prevalent in the population, to the point where that population now only express that type of allele. Here we say that the frequency of the allele now is 100% in that population. Because the frequency became 100% in that population, we say that the allele fixated in that population. So fixation describes the process through one allele is replaced completely by the other, and that allele no longer exists in the population. What you can observe here in this population at the end is that no genetic variation exists in this population. And now I ask you the question, is that good or bad for the population? Do you think it is good or bad that the population now only has individuals that look all the same? Think about the other evolutionary forces that we studied, especially natural selection. Is it good that only one gene fixates in that population? Probably not. So in our two next slides, I am going to discuss with you about the founder effect and the bottleneck effect. The founder effect literally means that one population migrates to some individuals of that population migrate to another environment, another territory. They carry their genes, and in that new territory they start a new population. Okay? So the founder effect happens when a small number of individuals in that population migrates to another area and they carry with them their gene pool. And this gene pool now becomes the new population, okay? Becomes present in the new population. This is very clear, for instance, when we look at the example on the right. Here we are analyzing our source population, the population that began everything. Here there are two alleles in that population, AB and AC. Here you can see, let's just assume that the allele AB happens in a higher frequency and that the allele AC happens in a lower frequency in that population. A certain group of individuals leave the area and migrate to another area where they did not exist before, just by random processes, okay? They could have been, for instance, separated by a tectonic event that literally separated those individuals, physically separated those individuals, okay? Or a mountain range, which is also involved with a tectonic event, appeared in that population. So the newly founded population, as you can see here, carries the gene pool from the source population. And in this new environment, the allele AC now starts to have a higher frequency. What can happen throughout time is that this newly founded population becomes a completely separated species, okay? This happens a lot in nature, that some random natural event happens separating those two populations, okay? That was also an observation that Darwin made when he went to the Galapagos Islands. He observed that the birds in the islands, they were similar but not identical to the ones on the on Earth, on the soil, not on the island, right? So he observed that even though they shared some similarities, they were not the same. So the new population that he was observing was somehow derived from the population from the land. That happened due to a probably tectonic event that separated those islands from the land, and that population became their own, they formed their own members, they formed their own new individuals. All right? So this is very common to observe in nature. Another event that happens in the genetic drift is the genetic bottleneck. Here the genetic bottleneck happens when a population, usually very large, is in a place, but in that place, something very drastic and extreme happens to that population that reduces that population to a very low number of individuals. Over time, that very little number of individuals might recover, they might produce new offsprings, but the diversity might be reduced. This is how we can observe that bottleneck effect. So we start with a population, with a random population, where we have different versions of that trait. And that trait can be literally anything. Then, after some time in that region, a very extreme and rapid environmental change happens to the population and kills a large part of these animals. But some of them still survived to that environmental change. This newly formed population still carries some genetic information from the source population, but as you can see, the genetic variability here now is reduced because now the trait is only being represented by two variations. This is also very common to observe when big events happens to those to the population of animals. Okay, I promise that our class is coming to a end. I understand that there is a lot of information to digest in this class, and especially because when we try to understand evolution and evolutionary mechanisms, those processes are things that we can't actually observe in a human time scale, right? Because those processes of, you know, natural selection, migration, or genetic drift, all and mutation, they happen in a much larger time scale that we can actually observe while we are alive. That's why we rely on the observation of fossils or other types of natural documents, okay, of those species. So I understand that sometimes that can be a little bit abstract because, again, we can't actually see. It's just like when you are studying biocell, right? And the professor tells you about the cell and all the things that's happening to the cells. Sometimes those concepts might be too abstract because we can't actually see those things with naked eyes. We would need, to go to the lab, perform experiments, use a microscope to observe those events happening in a cell. Here, we also need to use different tools to observe those evolutionary mechanisms taking place. Okay, so I truly understand and acknowledge that sometimes those mechanisms can be a little bit abstract from us to understand. But the main point of this class, and I know I've been saying that every single slide, but the main point that I wanted to understand is this process called speciation. As the name suggests, speciation is the process via which we can create two new species. Okay? Here we are going to see how species number 1 will give rise to species number 2 and species number 3. Those species that came from a source population, for them to be two different species, they must be isolated in terms of reproduction, meaning that breeding between between 2 and 3 can no longer occur. And if it occurs, does not produce a viable individual. Okay? That individual is not able to reproduce. So here, this is the main point of the class. Understanding that those evolutionary mechanisms can lead to speciation, can lead to the generation of different species. Okay? For instance, when we talked about migration, okay? Let's say that you have a piece of land right here, and then you have some islands right there. In this piece of land, you have your founder population. Some of these animals start migrating to other islands for different reasons. Over time, and I am even going to use other colors, over time, due to mutations, due to natural selections, due to pressures that are exclusive to each of these environments, the individuals become different. And let me represent here with different colors. They become so different to the point that they can no longer produce a new offspring. If these two organisms find each other, they can't reproduce and they can't generate a new individual, or an individual that is viable, meaning that the new individual is also reproductive active. Okay? These populations, were isolated from each other. And because of that, they grew up over generations. They grew up and they are now different species. Okay? So here we would have species number 1, number 2, number 3, number 4, and then number 5. Okay? What is important to acknowledge here is that even though they are all different species, they all came from the same ancestral, which is located right there. Okay? That is a very important misconception that sometimes us as biologists have to deal when we try to explain about evolution, right? Because people have this misconception about these ancestrals that they can't see, like, oh, how we came from apes or how we came from monkeys, because I can't see the ancestral for those two species, right? That's the thing. At the very beginning, we had one species that was ancestral to all of the primates, and then due to migration or genetic drift or even natural selection, those species of primates became so different from each other that they became their own independent species. Just because you don't see, and that's a very common misconception, just because you don't see a monkey becoming a human being doesn't mean that evolution is not happening, or that natural selection never happened, or mutation never happened. Okay? That is a very important misconception that has to be fought, because that's not how evolution occurs, right? That is the misconception associated with, for instance, Pokemon, Digimon, and all of those cartoons that we study, right? Speciation is a very important mechanism that will generate different species, okay? And that happens via reproductive isolation. What is driven the reproductive isolation is all those evolutionary mechanisms that we discussed. We've got to define, and I think I've already talked about this, that species are actually a group of organisms that they can interbreed, and they can produce a new offspring that can also interbreed. Okay? They can also produce new individuals. These individuals, they must be reproductively isolated from other groups. Because if they reproduce, then they belong to the same species. Again, that's a very general concept, and you might find in nature species that perhaps might not fit under this category. Okay? Speciation here is the process through which separate a single species into two or more species. All right? The process of speciation can happen through two processes. One is named anagenesis, and the other one is cladogenesis. Here I am giving you an example of a species of lizard. On y-axis, I am representing the form, which means the traits of those lizards. And here I am representing the time. Here at the beginning, I am showing you how species number 1 looks like. That species, for a very long period of time, will pass that period without actual changes, without profound changes. So throughout time, that species remain the same. However, due to a evolutionary mechanism, and that can be natural selection or that can be genetic drift, from that species something happens right there that you create a completely new species. And that can be, for instance, due to mutations that can happen throughout time, to the point that the mutations that occurred during this period caused these two individuals to be reproductively isolated, meaning that if they meet, they cannot produce a viable individual. The second process now is the cladogenesis. As you can observe in the cladogenesis, that species number 2 gives rise to two different species. Okay, that was the case for the primates, where we have a common ancestral that, due to processes such as natural selection, mutations, genetic drift, and migration, we have that separation into two new species. Let's think that, for instance, species number 2 was living in an area, and due to a tectonic movement or another natural phenomenon, now the area has a big mountain range that separated this land into two islands. Now the organisms in island number 1 and the organisms in island number 2, they no longer meet each other. Over time, small mutations and natural selection happen to these organisms that change them to the point that they are no longer the same species, meaning that if they meet each other, they are no longer able to produce a new individual, a new viable individual. Okay? We've got to understand that during this time, there are small changes occurring with that population. Okay? That at some point, those changes become so great that those individuals are not the same, and we categorize them into two different species. Okay? Like you said, sometimes it's hard to observe that, because these small mutations or this small natural selection that's happening throughout the time is not observable in our human lifespan. Okay, so that's why it's hard sometimes to understand that this is actually happening at all the times. Here, there are a few mechanisms that can lead to reproductive isolation. Okay, The main mechanisms is the occurrence of biological barriers that prevent or reduce the crossing, the interbreeding between those populations. That can be to a, for instance, ecological barrier, like the mountain range appearing in that island, or it can be to the changes in behaviors. For instance, you have a bird and for some reason they change their sexual behavior, and now those females are no longer attracted for certain males of that species, and now you lead to the separation, to the reproductive isolation of those birds. It can also be due to seasonal mechanisms, mechanical, or even physiological mechanisms. It is important that all those mechanisms will cause reproductive isolation. The reproductive isolation can also be divided into two isolating mechanisms, and I promise that this will be the last slide. We have pre-zygotic mechanisms and post-zygotic mechanisms to create reproductive isolation. Pre-zygotic isolating mechanisms, as the name suggests, before the formation of the zygote, which is the cell that comes from the combination of the sperm and the eggs. Here, the mechanism prevents mating from happening in that population because the individuals might not find each other, or they might not see each other as suitable mates. They might not find each other because there might be a biological barrier, or because they don't see each other as a suitable mates, like in the case of the birds, because there was a change in the behavior, in the reproductive behavior of those birds. Post-zygotic isolating mechanisms, like the name suggests, post is after the formation of the zygote. Here, isolation happens after the population mate with each other. So one happens before mating, the other one happens after mating. Okay? Again, I do understand that there is a lot of information to digest in this class, especially, can be a little bit difficult because, again, we can't actually observe that in our lifespan, but we can definitely know that this is happening through all those documentations in nature, for instance, due to the presence of fossils of those species that existed before the current species that we observe today. So go through those mechanisms. What I want you to understand is the main concepts about the population, how we understand the alleles, the frequency of alleles, and the frequency of the genotypes in that population, and also the evolutionary mechanisms in that population and how they are related with changes in the frequency of the alleles and in the frequency of the genotypes.